Okie dokie, so today we're talking about DNA discovery and kind of the race to figure out what was this idea of a thing that was transferring between living organisms and what passes on genetic code. So for the longest time, it was thought to be proteins because they had this code and there were 21 of them, so that seemed more likely. But from different sets of experiments, that was proven to be something else. So the first experiment is kind of Griffith's rats. And so what he did, he had some rough bacteria and smooth bacteria. Smooth. So I'm going to call that S bacteria. And so when you put the rough bacteria on it in a rat, the rat would live. So I'm going to put a smiley face. Smooth, the rats had pathogenic stuff casing around the bacteria, and so then the rat would die. Then he cooked the heated S. So he cooked the S bacteria and put that in, so to denature it and denature the bacteria, and that would make the rat live again. So then the rat lives. So then what he did is he applied some heated S bacteria plus some rough bacteria, and the result of the experiment was counter to what you would think, because we're adding a situation where the rat would live and a situation where the rat would live, and the rat actually died. So he described what he called a transforming factor. And this thing, something moved from the smooth bacteria to the rough bacteria that made them pathogenic, killing the rat. So the next another experiment that happened was Avery, McCarty, and McLeod. I spelled that wrong. Let's see, McCarty and McLeod. McLeod. And so what they did is they applied kind of a similar experiment, but they used RNase, DNase, and proteinase. And so they did this to kind of a similar thing like this. And then they did this, no change. And they did this with the protein, no change. But when they did it with the DNase, there was a change in what would happen, and the rats would go from dying to live. So rat die, put X, rat live, and then the rats would die. And so what was happening was these enzymes would break down the molecule, and so when you break down the DNA, the genetic material from the S can't go to the R, and so that means DNA is the thing transferring. So then you had the Hershey Chase experiment, and this one was more definitive. Hershey Chase. And this is kind of called the blender experiment. This is really cool. They used radioactive sulfur, so I'm going to put like a star. I'll put, like, what's the biohazard symbol? Like something like this. So they use radioactive sulfur, and radioactive phosphorus. And they added this to both the DNA of a T2 bacteriophage, so T2 bacteriophage. And so what the T2 did was it has this body and the legs, but it's only made out of two things. It's made out of like the protein outside, and then the genetic material on the inside. Let me get a different pen color for that. So genetic material on the inside. So DNA on the inside, and then protein on the outside. And so the sulfur was for the protein part, and the phosphorus was made for that. And so they had bacteriophages where they would be in this environment, and then they blended them up and noticed that, hey, the sulfur stayed at the top of the vial when they blended it up and sent it through a centrifuge, because all the bacteria would be down here. Bacteria. And then the T2. And so the T2 marked with sulfur is going to stay at the top. And so their radioactive stuff would be at the top. But with the phosphorus, they'd add it to the back. They'd grow the T2s in in the radioactive phosphorus. So then the radioactive phosphorus gets put in the DNA of the T2. Then the T2 would put their DNA into the bacteriophages they learned, and then they'd grind that up, centrifuge it out, and so you'd get, once again, a vial, the bacteria on bottom, but this time the bacteria would have their marking, and so T2, and then bacteria. And so the radioactive moved into the bacteria. So that was how they labeled that it was DNA moving from one thing to the other. And then, so that's part one. I'm going to go into part two and two more experiments that really cemented the structure and the discovery. So they discovered it was DNA moving through it that was transferring genetic information between things. So now, yeah.